So okay, so now we're gonna see so we were at homologous chromosomes. So here's another part where students really get confused with. So homologous chromosomes, it's important to know what this means. Homologous chromosomes are essentially just chromosomes that are the same size, shape, and contain the same genes. This is very important right here. Okay, so remember how I said 23 of your chromosomes from your mom, 23 comes from your dad. In all 23 of those chromosomes, we have essentially the genes that we need in order to survive. So, for example, remember how there's 23 pairs of chromosomes, right? I shouldn't have used blue for this. There we go. Okay, so there's 23 pairs of chromosomes, and it should technically be called 23 homologous pairs. There's kind of like a hidden connotation there, but essentially it means 23 homologous pairs, which means there's 46 in total. Okay, now I'm gonna draw only 3 pairs out of the 23 pairs, because drawing 23 pairs would be too much, but essentially chromosomes that look the same, they have the same size, shape, and contain the same genes, pair up together during meiosis. And I'll go through the phases real quick. Let's make this one very tall. There we go. So I purposely made the second chromosome small to show that it has its own homologous chromosomes. See how they're the same shape, they're the same size, and they most likely contain the same genes as well. So I've only drawn out 3 out of 23 pairs. Now in humans, there should be 23 of these in total, 23 pairs. In each pair, they contain the same genes. Now this one is important to understand. For example, eye color. Let's say that these 2 chromosomes here contain eye color. Remember, you get 1 set from your mom, 1 set from your dad. So there's 2 copies of each gene in our body. So for example, for the gene of eye color, we have 2 copies. 1 from mom and 1 from dad. So that's why you kind of, for example, if your mom either has blue eyes and your dad has brown eyes, then, and if you have brown eyes, how you have 2 copies. So the copy that the eye color that your mom gave you, so I'm just going to draw this out actually. So for example, let's say your mom has blue eyes and let's say your dad had brown eyes. Essentially, they each give 1, right? So essentially what I'm talking about is the mom, this side, the dad, this one. So they're homologous because they contain the same gene, and in this case we're talking about eye color. And then you essentially get 2 copies of the eye color gene. So you have blue, the blue eye gene, and there's the brown eye gene. So we're gonna learn about this in chapter 18, but essentially brown eye gene is dominant, which means that if you get this copy, this is what you're gonna this is what's going to appear. So in your body, remember your 2N, you have 2 sets of chromosomes, you're diploid. You have 2 copies of it. It's just that only this one is expressed, essentially. You have 2 copies of the eye gene. You have from the blue eyes from your mom, brown eyes from your dad. But this one is the only one that's expressed. So always remember, for each gene, there's 2 copies, and this is essentially the whole concept of homologous pairs. So the chromosomes that contain the same genes, they line up together, and that's what homologous chromosomes do. That's what homologous chromosomes are. They contain the same genes. There's 2 sets of it, essentially. I just used eye color as an example. Okay, now that we know that knowledge, let's look through each phase of meiosis. So first off, we have meiosis 1. Let's go through meiosis 1. And meiosis starts with prophase. Specifically prophase 1. I'm just going to write prophase 1. Now, same thing with mitosis. Prophase, chromosomes condensely, packed tight together. Nuclear embryo starts to disintegrate, centriole moves to pulse, spindle fibers are formed. So all those things are happening basically. So I'm going to draw that out. I'm going to draw out that the nuclear envelope is disintegrating right here, and that the centrioles are going to move to the poles. There we go. Yeah, it's still off center, which is good. You don't want it to be fully in the center yet. And the spindle fibers are starting to form as well. So these 3 things are the exact same as mitosis. Main difference is that crossing over happens. Actually, I'm gonna do this in sequence. What happens is pairing up happens. Pairing of homologous chromosomes happen. So remember, I used the eye color gene example. So essentially, the chromosome that has the eye color gene from your mom, the chromosome that has the eye color gene from your dad, they're gonna pair up together. For example, there's another chromosome that has to do with your let's see, your earlobe or something, your ear shape. That chromosome from your mom is going to pair up with the chromosome that has to do with the ear shape from your dad as well. So obviously there's 23 pairs of that in humans, but for convenience sake, I'm just going to do 3 pairs, which is essentially just 2n equals 6. If there's total of 6 chromosomes, that means there should be 3 pairs of them. But they come from 2 parents, right? Because it's diploid from 2 sets of chromosomes. Okay, so I'm going to show those pairing up together. So I'm going to show one from your mom, one from your dad. So say that this one is your, I don't know, eye color, right? And I used an example about earlobes, maybe that. Maybe this one is how much, maybe this one is about widow speak, if you have a widow speak or not. So we're going to make this one like that. And notice how they're the same shape too. So this one is the gene of the widow speak from your dad. This one's the gene with your eye color. And this one's your ear shape. So they pairing up together, and that whole pairing up process, it's what's called synapsis. So the process of pairing together is called synapsis, and what they do when they pair up is they share genetic information. So what that's called is crossing over. So crossing over is sharing of genetic information. This is important. This is one of the things that's important for variation in gametes. So I'm going to actually highlight this because there are 3 ways that you can get variation in your gametes, and this is one of them. So they're pairing up, right? When you look closer, I'm just going to like do a zoom in visual here. So this is a zoomed in version of these 2. What's actually happening is that they're exchanging information with each other. So I'm going to draw something like this. There we go. So something like that. Okay, and then for the dads, so they're kind of just like shaking hands essentially. That's what's happening if I use that as an analogy. What's happening is they're shaking hands here. So see, they're exchanging information. That process is crossing over. Now there's another term. So first off, we've been introduced to three terms so far. There's synapsis, which is the process of pairing of homologous chromosomes. There's crossing over, which is when they share genetic information. And then there's what's called a chiasma, which is the site of crossing over. So that's just the site where they cross over. It's called a chiasma. There you go. So when that happens, what ends up happening when they share genetic information is that some of your father's genes can go to your mom's chromosome. Some of your mother's maternal chromosomes can go to the paternal chromosomes, and they can end up looking like this. Just doing. So let's say some of the maternal chromosomes got it wouldn't be there, around right here. So see how where they shake hands, so they exchange chromosomes there, and that's what causes some variation. So it's now mixed up together. Some of the paternal chromosomes are in the maternal chromosome and vice versa. Okay, so that's the main kind of thing that happens in prophase 1. Pairing up, crossing over, side of crossing over chiasma. Some of the chromosomes get mixed up. Basically, they change genetic info, or they exchange contacts. Okay, now metaphase 1. Same thing with with mitosis. They line up in the middle. Metaphase 1. There we go. Line up in middle. Okay, here is the big distinction. The homologous pairs line up. Pairs line up together. So you see how in mitosis 1 in metaphase, where they're all lined up in a single file, where in meiosis 1 the homologous chromosomes are lined up side by side. So see how these two are homologous but they're not lined up side by side. These two are homologous but they're not lined up side by side. They're all lined up in the middle. But so essentially, because they all line up in a single file, when they get separated here, you still have two sets of chromosomes, right? One from your dad, one from your mom, one from your dad, one from mom. You still have two sets. For example, if that's the I gene, I color gene, you still have two sets of it. You still have two sets of it, of the ear gene, and then you still have two sets of your widow's peak gene, because of how it's lined up in mitosis. But in meiosis 1, that's the big difference, and we'll see in a bit how that affects the chromosome number. So metaphase 1, homologous pairs line up together. So I'm going to draw that out. And same thing with mitosis. The nuclear envelope has formed, centrioles are in the poles, spindle fibers have attached to the homologous chromosomes. Okay, so I'm just going to write this out real quick. And this one. Now I purposely laid it out this way just to show you that it doesn't matter where the paternal or maternal chromosomes line up. So they could go to the left side, they could go to the right side, it doesn't matter. And that actually causes variation as well. But before that, I'm just gonna draw out, I'm gonna show that crossing over happened in the prophase, showing that some of the paternal chromosomes got here. So I'm gonna do that. You should add this much detail in your exams as well, showing that crossing over happened in each one. And remember how I told you that it doesn't matter whether the maternal or paternal chromosomes are on the left or right side? That's what's called independent assortment. So this is your second cause of variation in gametes. And if you think about it, in humans, we have 23 pairs, right? Which means that there's many combinations of how this could happen, which leads to variation. So what could happen is, this is just a possibility, what could happen is that the paternal chromosomes are all on the left side, which is obviously, it's a very low chance. Maternal chromosomes on the right side, or it could shuffle either way. That's what's called independent assortment. So there's no more nuclear envelope. You know, I'm just gonna adjust the centrioles black now, and the spindle fibers. No, I'll make the spindle fibers orange. There you go. There we go. So it's connected here. There we go. Independent assortment happens here. Then homologous pairs line up. And when they do that, in anaphase 1, homologous chromosomes are separated, not the sister chromatids. Are separated. So see how in here, when they separate, because they're lined up all in the middle like that, the chromatids are separated. So essentially you're not losing genetic information because this copy is the same copy as that one, this copy is same as copy as that one. But in here, when you separate the homologous chromosomes, there's only one copy of each gene now. So I'm just gonna draw it out. First, there we go. I'm gonna do this. I'm gonna show that each one is pulled away. There we go. This one's, yeah, there we go. This one kind of looks like that. Okay, and then, okay. I'm gonna draw this out, just showing again that there's already variation there due to crossing over. Okay, this one. Okay. So remember how these two are the genes for, because remember the homologous chromosomes pair up together are genes for eye color. This one, these two are the genes for what did I say earlier? Ears. So this is just examples, by the way. I'm just showing examples that this is the eye color gene, ear gene, but obviously it's more complicated than that. This one's the gene for widow speak. So see how we had two sets of it, one from your mom, one from your dad. Well, when you separate them together, we each only have one pair now. One set of each gene, which means that a telophase. Oh, actually, I'm gonna draw this out too. So in anaphase, when they are segregated together, that's also a cause of variation, because when you separate them together, they're gonna be in different cells. They're gonna be in different gamete cells, which means that segregation, or the separation of these two homologous chromosomes, is another cause of variation. So what I highlighted here, these are three important things. These are what causes variation in each gamete. And so in telophase, I'm just going to draw a telophase. Here we go, telophase. In telophase, essentially, same thing. Cell starts to elongate. Nuclear envelope is forming around each one. The spindle fiber.
Oh yeah?
Yep. Sorry about that. Okay, so we were in telophase now. So the homologous chromosomes have separated, which means there's one set of genes in each cell now. So let's draw it out. The nuclear envelope has been formed. In this cell, we have this chromosome right here. We have another one. And then we're just going to list out. There's these small amounts of things. And there's also the paternal chromosome with a bit of the maternal chromosome due to crossover. And this side, same thing. I'm just gonna draw these out real quick. Okay, so there we go. Hold up. Okay, so now I drew it out. So remember how we used to have two sets of each genes, which means we're diploid. Now we only have one set of each gene now in meiosis. So here, remember this one is the eye color gene. So in each cell, we only have one gene for eye color now. Only one gene for your ear shape. And only one gene of your widow speak gene. So because of that, because we go from two sets to only one set, After meiosis 1, we end up to 2n to n, which means we go from a diploid organism, we have 2 sets of each gene or chromosome, to haploid, which means you only have 1 set of each gene. Does that make sense? Okay, so telophase, these things happen. And cytokinesis, it happens in between meiosis 1 and meiosis 2, cytokinesis. Too lazy to write it out, but essentially, yeah, it's like, hold up, it's like this. So now we have 2 cells, the cells have split off. Each one now has 3 chromosomes. Right here, there we go, this one right there. And same thing in this side. Okay, and at the end of meiosis 1, we should have n equals 3 in each cell, which means that the number of chromosomes has dropped, and also the number of ploidy. So we go from diploid to haploid after meiosis 1, and we get from 6 chromosomes to 3 chromosomes. But we can still go through meiosis 2, because remember, we can split up the chromatids as we did in mitosis here. We split up the chromatids, which maintains the same number of chromosomes, because when we split it up, remember, if we split it up during anaphase, so this is just a quick kind of example. If we split this up during anaphase, these are spindle fibers, then each side is gonna have 1 chromatid. So remember how this one is 1 chromosome. If this goes into 1 cell, then this one becomes 1 chromosome as well. 1 chromosome, this is 1 chromosome. So see how number of chromosome is maintained, but the number of chromatids have halved. So in here we had 2 chromatids. Now in each cell we are gonna have 1 chromatid. Okay, now we're gonna go through how that actually happens. So meiosis 2 starts out with this, essentially. So we got this now. The main difference between meiosis 1 and meiosis 2 is all of these crossing over, independent assortment and segregation doesn't happen anymore. So essentially what meiosis 2 is it's very similar to mitosis. And we will see that here. Okay. Meiosis two. Remember, this is very similar to mitosis. Okay, so we're gonna just draw out both of the cells. So at the end of meiosis 1, we ended up with 2 cells, each with n equals 3. Can I copy? Paste. Here we go. And then this side as well. Copy. There. So we started off with this in prophase. So we're gonna write this out as prophase. But remember, in prophase, it's the nuclear envelope is starting to disappear. So I'm gonna draw it out as it looks like it's breaking. Oh, and there's something that I actually forgot here in the cell. Remember, your your centrioles should always be shown. Just gonna draw it as black. But in telophase, there's gonna be no spindle fibers. So at the end, there should still be centrioles. And I'm gonna draw the centrioles here as well. It's starting to go to the post in prophase 2. Spindle fibers are starting to form. So I'll draw it out like that. So in each cell, we're now at n equals 3. And just for clarity's sake, we're gonna draw out. I'm just gonna write out how many chromatids there are. So in each cell, if you count out how many copies there are, so there's 1, 2, 3, 4, 5, 6. There should be 6 chromatids, but 3 chromosomes. So same thing here, 6 chromatids, 3 chromosomes. Okay, so that's what happens in prophase. There's going to be no more crossing over, because remember in prophase 1 it was pairing of homologous chromosomes, but it has no more homologous chromosomes because there's only one set of each gene now, which means that all of these should be its own gene. Alright, metaphase 2. Metaphase 2 should be the exact same thing as mitosis. Okay, so now there's going to be no more nuclear envelope, and I'm just going to draw out all 3 of these. Okay, and then the chromosomes. The big difference with metaphase 2 and metaphase 1 is metaphase 1, the homologous chromosomes line up side by side, but in here the chromosomes line up at the center like in mitosis. And when that happens, the chromatids are split apart instead of the homologous chromosomes. But as you can tell, there's going to be no more homologous chromosomes in each cell, right? Because they've been separated away in meiosis 1. So that's why they are shaped like this. Okay, so spindle fibers attaches to each one. There we go. Blah, blah. Okay, so same thing in meiosis. Essentially, mitosis. The chromatids are split apart. I'm just gonna speedrun it at this point because I feel like it's kind of self-explanatory, I'd say. Okay, just drawing out the centrioles, spindle fibers. This one should have one chromatid because the chromatids are not split apart. This one, and this one, this one, and this one. Obviously, it's all mixed together now, right? So some parts are blue from crossing over at meiosis 1. Yeah, I'm just there. So anaphase 2, and as you can already kind of tell, each cell is still going to have 3 chromosomes, but there's now going to be 3 chromatids in each one. Telophase 2, same thing, each cell is going to start to elongate. The spindle fibers are gone now. The nuclear envelope is going to start to reform. So I'm just going to draw it. Nuclear envelope starts to reform. Spindle fibers gone. In each cell, there should be 3 chromosomes still. But as we know, it's not really depicted well here, but each chromosome should have stuff mixed in now. There we go. So now in cytokinesis, that's when the cytoplasm splits off into 4 different cells. Cytokinesis is when the cytoplasm splits into 2. You're now going to have 4 daughter cells, which I have talked about is the product of meiosis. Why did I put this? Is supposed to say cytokinesis 2. Actually, it's not called cytokinesis 2 because cytokinesis isn't part of meiosis, it's only PMAT. So this is going to be 2 cytokinesis. Here we go. Okay, so now this should be its own separate thing. Okay, so now each cell should be a nuclear envelope there now. Centriole, 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 centrioles, 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 and each one gets 3. Ta-da, ta-da. Obviously, for humans, each one gets 23, right? I just used 2n equals 6 as a starting cell, just because it's easier to draw out than having to do all 23. But again, it's not clearly shown, but each cell should have a bit of both mixed in due to crossing over. That's what I'm doing right now. There we go. So, if you count it, in each cell, which is our gametes, there's 3 centromeres, which means there's 3 chromosomes. And it's now n, because remember, we only have 1 set of gene for each cell now, not 2. N equals 3, n equals 3, n equals 3. So we have the same chromosomes as we had at the end of meiosis 1, n equals 3. The main difference is that this has 6 chromatids, remember 6 copies, but now there's 3 chromatids at the end here. So there's 3 chromosomes in each, zomes in each, and 3 chromatids in each. And it's now haploid. So after meiosis 1, it's haploid, and in here it's still a haploid. Haploid. There we go. There we go. So I hope that clears some stuff out. Just for a summary of meiosis. Meiosis to after the end of meiosis 1, we went from 2n equals 6 to n equals 3 in 2 cells. So remember, after meiosis 1, it ends up becoming 2 cells. 1 cell. After meiosis 2, and it's still n equals 3 now in 4 daughter cells. But when we count the chromatids in here, we would see that it started out with 12 chromatids if we count out all the copies. So again, if we just like look through here, there's 12, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, because they're duplicated chromosomes at the start of meiosis and mitosis. But in here we have, so our chromosomes became half, so it's 3 chromosomes now. But they're still duplicated chromosomes, which means there should be 6 chromatids if you count it. So at the end of meiosis 1, if we see, where was it? Yeah, as we see here, see how each one has only 6, 1, 2, 3, 4, 5, 6. Here there's 1, 2, 3, 4, 5, 6. And here, number of chromosomes still the same, because remember, it all depends on the number of centromeres. So these two are 1 chromosome. It's just the number of copies or chromatid changes. So in here now it's 3 chromatids. So that's how this all changes in meiosis. For mitosis, we started out with 2n equals 6, same thing. But mitosis end, it's still 2n equals 6, but now it started out with the chromatid started out as 12. But at the end of mitosis, the chromatids changed to 6. Because see, there's 1, 2, 3, 4, 5, 6 copies, 6 chromosomes. But in here, we started out here. There's still 6 chromosomes because there's 6 centromeres, but there's 12 copies. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. So I hope that clears some stuff up. Make sure to watch this before, let's say, before the weekend, I'd say. I feel like at this point you should really be familiar with mitosis and meiosis, each steps, how many chromosomes are in each one. I know this is a very hard topic, but once you get this, you'll never forget it. And yeah, if you understand this, you basically kind of got through the hardest part of Bio 30, in my opinion, and of many too. So yeah, that's it.